Good morning everybody. So, in this last class of this particular course where we will be talking about the sensors, we will so finish first those about the sensors and where we are trying to handle or tackle the any sample, whether you have a polymeric sample or a water sample or any environmental sample containing the phenol group. So, to how to sense that, but the basic idea behind all these things whenever we try to develop that sensing mechanism is that what are the molecules you are looking for, because in this particular class also we will be talking something how you define your problem, how you. So, first thing that you have to define your problem, how you define that particular problem and what are the methodologies you can follow, so that you can identify, that means initially detect and then quantify the total amount of the sample present in it. So, right now we will finish this and then we will just consider a very simple example, because this is a very good source of metal ion, and if we can track that, how to track the calcium in diverse varieties of samples and what particular methodology you can follow, and depending upon the very important thing, the concentration range. That is why if some methodology for your analytical chemistry is failed, we should go for the next one, and as we go down in all these concentrations, we should be very much careful, starting from the sample, getting the sample from the real world to finish the experiment and the data and the percentage and reporting. So, what we are talking in our last class, that the phenolic compounds and their presence in different species, and we just basically go for a oxidase type of enzyme. So, is the oxidase, so is the tyrosinase, because the name will also tell you that is related to the tyrosine, which is an amino acid. So, if that amino acid tyrosine has the phenol unit itself, so inbuilt phenol unit is there in the tyrosine amino acid. So, copper will be required for this particular type of monophenol monooxygenase reaction, and it has been identified in the biological world that this phenol can be oxidized by this copper. So, understanding everything from that biological world is also very important, because once you know these things and if you critically see that okay, that amino acid, it can be a, obviously it is not a separate amino acid, but it will be a part of the polypeptide chain or any other substrate containing the phenol group. So, whenever you have the phenol group, that phenol group can show some kind of activity, and that basically give rise to the corresponding electron transfer reaction, because if this is there, so we know like your water molecule, you have two lone pair of electrons on this phenol unit, and when it is going for the deprotonated form, that is the phenolate ion, it can form a coordinate bond to a copper 2 center. So, if we go for some electron transfer on this copper 2 plus system, and if we can go for some other type of reaction, that means whether we can go for activation of this CH bond, because all of them are CH, you have 6 CH bond in the benzene ring, and so likewise you have 6 instead of 6 you have 5, this is 1, this is 1, this is 1. So, all 5 CH bond can be activated to convert this CH to COH. So, is a typical hydroxylation reaction. So, is a copper catalyzed hydroxylation reaction on the phenol group, and why we require this particular phenol group, because the phenol can approach this copper center through this activity, that means the weak interaction and ultimately the coordination bond which is forming between oxygen and copper can give rise to this sort of reaction. So, whenever you read it this sort of thing, you should also think of why this particular reaction is going on. So, these two reactions, even if you consider that the first one is the phenol, and when you have the monooxygenase, you go for a diphenol reaction. So, half of this O2 molecule. So, how much O2 molecule is being consumed, as we have seen earlier, that we can have some development of the presence of O2 by looking at the corresponding O2 sensor, that means the oxygen sensor. So, here if we can have some monitoring process or monitoring mechanism, where we can see that this particular O2 is consumed, and if the concentration is going down, is decaying. So, the amount of oxygen being consumed for this particular reaction, we can consider that particular reaction for its monooxygenase reaction. So, is the monophenol oxygenase reaction means that if you can have, even if you have this particular phenol group, and if you go this one, because this you can get from the simple benzene ring. So, the benzene ring if you have, you can go for the corresponding hydroxylation reaction. So, is your hydroxylation reaction, and this hydroxylation reaction is going to make you that CH to COH. So, once you have this, that means your phenol is there. Now, it is activated by the copper also, because this can also be activated by copper or any other metal ions. So, if you have this, so this phenol unit is there. So, where the second OH function can come, you can have, you know that the options you have the ortho position, the meta position and the para position, but since it is OH that copper activated process, that means copper should be nearby. So, only these two positions, that means the ortho positions will be activated and you get that diphenol. So, is therefore, it is ortho diphenol what you will get. So, this is basically the corresponding oxygenase reaction, and this basically this ortho diphenol is another name of this is the catechol. Why these are important? Because we know that the several neurotransmitters are also available in our body, and catechol is also very good molecule for your potato peel also, that it is browning. When you know that the potato peel is there, if you remove the potato cover skin, it again immediately goes for the corresponding browning. So, the browning of the potato cover or the potato skin is due to the corresponding catechol formation, because it is highly intense in color. When you go for this, so something else unlike your this particular monooxygenase reaction, since it is working on catechol, it would be called as the corresponding catecholase. So, this catecholase will now work on it. So, what will happen, whether some other modification and all these things, because this is basically the insertion of oxygen what we are getting from half O2. So, this is basically going for this half O2, O2 is being inserted over here. So, this CH will be COH. Now, what will happen on this? So, this will not go for any kind of the other third oxygen insertion on this, but it can go for its typical oxidation to its orthobenzoquinone. So, is a orthoquinone or benzoquinone we call, and is a strong optical absorption it can show. So, the darkening of the color and the strong optical absorption you can measure, and you can find that there is a corresponding reaction for this particular one. So, this particular reaction can take place. So, this H will go. So, again half of this O2 can consume to give you the corresponding H2O. So, is basically these two reactions are one after another can take place. So, the diphenol reaction for formation of the diphenol as well as the quinone formation, this is simply the corresponding electron transfer reaction. Two electron transfer of this catechol. Catechol is going to first to semiquinone and then semiquinone to this orthoquinone. So, if these two reactions are happening and we have to monitor the corresponding presence of phenol or the corresponding presence of diphenol in the medium. So, what we can do? If you have the phenol oxidase in your hand, and that phenol oxidase can give rise to not only the biological thing what has been discovered earlier for your orthodiphenol unit, but it can also give you the corresponding para-benzoquinone formation, because this para-benzoquinone formation is also an interesting biochemical reaction which is forming from the diphenol, which is para-diphenol, and which is immediately forming, and there also the corresponding darkening in the color, but the structure is wrong here, it is taken from some paper somewhere. So, this you have the double bond here and you have the double bond over there. So, this particular one like this, you have the corresponding conjugation of the double bond here, here, here. So, it is also colored. So, this coloring mechanism you have, and then this thing that whenever you do this reaction, whether it is your oxidase reaction or your catechol oxidase also, the catecholase reaction, what is happening there? Now you are consuming this O2, because earlier we have seen that O2 we can determine from Clark oxygen electrode. If you have the Clark oxygen electrode, the O2 concentration in this enclosed form or the enclosed area can be determined. Now, if this O2 is being consumed by the phenol oxidase and forming your water molecule, so the reduction in this O2 concentration as well as the formation of this quinone. So, quinone formation you can monitor, and the reduction in this O2 you can also monitor. So, these two monitoring process independently can give rise to the corresponding measurement, that means oxygen consumption measurement during the phenol oxidation with the phenol oxidizing enzyme. So, if you have the phenol oxidizing enzyme, so consumption of O2 you can monitor, or otherwise you can detect the corresponding concentration of the quinone formation in the reaction medium. So, this basically gives rise to the corresponding formation of these, and when we develop these, likewise that of what we developed for your oxygen sensor or the glucose sensor. So, definitely we should get some applications of these phenolic sensors. So, now we slowly move to in this particular last class basically. So, initially we will just talk about the applications of the sensors and in broader respect to the electrochemistry, and then how we handle these applications for some real world sample. If we have any real world sample in hand, how we can monitor that particular one. So, analysis of real samples, which is also a practical aspects of your analytical chemistry. So, for the phenolic biosensors, so every chapter will have, whenever you develop something, whenever you know something, always you should think of the applications, because it is a typically a applied science. So, applications is the first and foremost thing. That is why we are one time we are comparing that who is your analytical chemist and chemical analyst. So, analytical chemist will give the procedure of these measurements or the analysis, and when the application is going on, there is no need to develop that particular process and that particular point, only how many samples you can handle, how many samples you can determine for their phenolic presence. So, is very much required for environmental control. So, we will just talk about 3 such applications, but carefully you listen these, that these are very important things, that how you can think of a subject where we are talking about so many theoretical things like that of your Faraday's current and the voltaic current and all these electron transfer and all these, but ultimately how it is applicable to an environmental control. That means that when in the countries there you have some strict environmental restrictions, you cannot go beyond to that particular level. So, first thing will come is the monitoring of your wastewater. You have to treat the wastewater, then only you can dump it, you can pass out that water in the drain line. So, the whatever water you are discarding, even from our laboratories also, for the laboratories like us also, the academic institutions, not the industrial institutions or any R&D laboratory, we use very small concentration of these materials for our teaching purpose and little bit of our research purpose, but we should not dump that water, because we know that water is very much contaminated, and if it goes and if proper care is not taken, it goes through some open land or open drain. So, it goes down to your groundwater. So, groundwater will also be contaminated. If your drain water is contaminated with phenol, your groundwater at one point of time will definitely be contaminated by your phenol. So, let us see how we can detect that. So, for environmental control, the phenolic biosensors will be very much useful for that purpose, for the detection of the contaminants on surface as well as groundwater. So, as you see here also, that surface water, your lake water, your river water, and the groundwater is also getting contaminated. So, several tyrosinases you have, then the corresponding GDH, so any dehydrogenase and CDH based biosensors. So you just don't bother about these terms, basically the quinone based or some other this reduced form of these based biosensors proved useful for assaying a phenol index in water. Is a terminology, is very useful terminology, that phenol should be avoided in your water, even for any kind of water. It may not be your drinking water. So you have the phenol index in water on surface river water during field test. So you take the sample. So if the biosensor what you have developed is a very small one, and if you can take these to the field, so immediately in-site determination of phenol is possible. So if it is like handheld pH meter, if it is some handheld pH meter, you can take that and the electrode can be inserted on the water, whatever water is available over there in the field, and you can tell us about the amount of phenol present in that particular water. Then a broad application will be in the area of pharmaceutical industry, medicine and biotechnology, because just now we are discussing about the catechol oxidase. So some molecules are also known as catechol amines, and they are good neurotransmitters for our neural activations, for our neural signaling transfer. So you see that the name will immediately tell you, you know the amines like ammonia, so organic amines you know. So catechol bound to some amine part. So you have to have some part where you have the possibility that amine can be oxidized, the amine part can be oxidized in presence of your oxygen, and catechol can also be oxidized, as we have just now we have seen that you need the enzyme catechol oxidase, and if that enzyme is available and if you supply the required number of electrons from the electrode, it can give rise to the corresponding biosensors for not only for your phenol, but also for your catechol and again for your catechol amines. So when these are present for these diverse type of samples, so pathologists, the clinical analytical chemists, they also handle these blood samples, the brain samples, the brain whatever sample you can take out from the brain, and also the cell cultures, and for the detection of cell numbers and sensitive lab detection in enzyme immunoassay. So enzyme immunoassay, if the enzyme is that which is a catechol oxidase type of enzyme, that can be assayed very nicely by knowing the corresponding information of your phenol biosensor. Then large number of food material can have the corresponding species as your phenol species in it, like your olive oil, red wine and milk product. Already I told you in our last class also. Now through this biosensor, if the concentration is very less, say some microgram or nanogram level or the ppb or the ppm level, parts per billion or parts per million level, so this can also be detected for assuring your food quality that no, it has not been contaminated much with the level of phenol present in it with respect to your type, that means whether you have a olive oil sample or a red oil sample or the milk product sample. So whenever you read all these things, all these informations you can gather from any book or any research article, you should always have some idea how it is being detected. So what sort of analytical chemistry is involved over there, and why we are only bothering to identify these, because these are very important samples and very important real life samples in our world. So now we just move on to the overall applications, and if we just go for the overall applications, so which is very important now, that applications we are seeing for electrochemistry or the bioelectrochemistry or development of the sensors. Now when we talk about these overall applications, and we have to know that we go for analysis from the help of analytical chemistry. So this analysis and analytical chemistry, so initially we will take a bit of knowledge, and a knowledge is required, the chemical knowledge or the chemistry knowledge is required But if we can apply this for any other areas, all other areas, so is a basically it can be considered as a science. It should not be restricted within chemistry, because you start with chemistry, but when you are ending up with something, it is covering all the different areas. So, any analytical chemistry you open up, you will find there are large number of areas which it can cover and which it can go for its applications. So, is centrally you can have the corresponding analytical chemistry, but when you apply it to get some analysis, suppose if you have a cement sample in one hand and another hand you have the blood sample. So, how you correlate these things, how your knowledge of analytical science can be helpful to identify one of the component, that means you are not analyzing, that means some of these we consider as the total analysis, and sometime we will go for only the presence of Fe2O3, the amount of iron, because iron is not wanted to be present in good quality of cement for building material as a good building material. So, if you see that even if you do not know the area where you are, because this cement you know the chemists are handling, the civil engineers are handling, the architects are handling, and people who are making this also in the industry, they are also handling this cement. So, they should know the purity of this material and whether you have the permissible amount of Fe2O3 in it, and sometimes we can go for the total analysis for all these things. So, if you go for this overall application of this, what we get the exact information about the composition and structure of the matter. So, initially you get the composition, that means the percentages we require to know. So, we get what is that we go for the analysis, this analysis, again we are talking about the analysis. And suppose we have a small organic molecule we have prepared in the laboratory. So, what you can go for the first analytical chemistry you can apply to know this analytical molecule, that molecule, organic molecule. So, we apply different spectroscopic techniques to little bit understanding about its structure and its bonding and all these things. But the very basic thing is we can do, because these are very small amounts, so only few milligrams will be available. So, 1 to 4 milligram if they are available for analysis, milligram if they are available for analysis. So, what you get that we can go for a micro analysis, and earlier we are knowing only that if your organic molecule, we all know that organic molecule is forming with carbon, hydrogen, oxygen. You can have also nitrogen, you can have also phosphorus, and you can have halogens. So, these all are there. So, in the laboratory also, when the amount is more, you know from your school days that you can analyze this corresponding organic molecule for its presence of nitrogen and presence of halogen also. The first thing we know, we learn it from our school days, that in organic molecule how to detect the nitrogen and the halogen. But if you go for the micro analysis, and which is automated one, so it gives rise to the percentages of, initially we call it also the CHN analyzer. So, the PerkinElmer or any other instrument making companies, they are giving us this CHN analyzer. So, CHN analyzers, immediately if you go for this particular analysis, it can tell you the percentage of carbon, percentage of hydrogen and percentage of nitrogen, again taking sample in the range of 1 to 4 milligram, which is not possible to detect conventionally with a sample of this range for the percentage analysis of all three together. So, micro analysis is very important with regard to this particular one. So, very basic thing what we can use is the analysis of any molecule containing carbon, hydrogen, oxygen, nitrogen and all this. So, if you go from here, that means if you, instead of this cement sample, any material you have also, and once you know that the material has oxygen, you know. So, you can detect that also, that particular presence of oxygen in that particular material. So, the composition and the structure, because the spectroscopic techniques will tell you structure, if you have the double bonds, how many double bonds are there, if you have ethylene sample in hand, if you have the polyethylene sample in hand, and if you have also the living world sample like beta carotene in your hand. So, how you can detect all these things in terms of the corresponding values in the lambda max values and the epsilon max values. So, this is together, both these are together, that means it is a art, that means you should be mastered enough. So, it is an art and science. So, what scientific knowledge you must have to deal with this analysis is that a very little amount of chemical knowledge or the chemistry knowledge you must have, but basically it is a typical art, that how you develop this thing, that means with your knowledge, that means some amount of knowledge you must have. So, if we just side by side, if we see philosophically what is the philosophy behind all these things, is that if you have this particular one in terms of your, why we are talking it as art and science. So, if you have this particular one, that means you must require some knowledge, and knowledge, what we know, that this particular knowledge what you can gather from the chemistry, because it starts with the chemistry, finding out of whether you have nitrogen in any sample or halogen in any organic sample. So, this chemical knowledge is there, then the problem will come, that you have to define the problem. How you define the problem? That means whether you are trying to determine the development of the sensors, like development of sensor for O2 determination. So, you define the problem, then you try to solve it in a way that how you can get this particular information related to this O2 concentration. Then that means your intuition should be applied, that how you tackle this thing, how you apply your knowledge to get this thing. And once you do, it is not that only one sample you analyze and you get good results, even if you handle instruments, that means it can be simple instrumental methods of analysis. So, you must be experienced enough. So, your experience is important. So, scientific part is related to your knowledge, and your this experience and mastering of this thing will be related to your art of this particular subject. So, you have to be mastering yourself for do all these things. So, that is why we can consider it as the art and science of determining what matter is and how much it exists in that particular sample. And also it has applications including in forensic science, bioanalysis, clinical analysis, environmental analysis and material analysis. As I told and repeatedly say every day almost, that what are the areas you can handle. So, use their knowledge of chemistry, that is why we are using your knowledge of chemistry. First you start with that particular knowledge, then the instrumentation. If we consider that okay, I cannot handle by simple burette pipette analysis or titration or the gravimetric estimation, you have to go for the instrumentation. So, what instrumentation you should need, whether you require a visible spectrophotometer or a gas chromatograph or a mass spectrometer. Then some small amount of knowledge you require for handling those computers, because most of these instruments are now software driven. So, how to run those softwares and how to run the protocol for these, because the computer will acquire these things. Like your cyclic voltammetry is also your computer, you will see in the screen, but how to store the data. So, not only the image what you are storing, but also the data you have to store it, and the statistics, because the analysis you have to do on those data. That means, when you go for the corresponding use of simulation packages, as I told you earlier in one time, that simulation packages are there and it should be with you, such that you can extract out some good information from that particular spectrum or the particular voltammogram. When you handle the voltammogram, you know all the data including your kinetic parameters, you can extract out from the line shape of the voltammogram. To require these statistics to solve the problem in almost all areas of chemistry, forget about chemistry. So, in all areas of science also, several scientific areas you can handle, even you can go for some analysis of the art object, analysis of the historical objects what has been found from the earth, and all kind of industries can be served with this particular information and the knowledge. So, Their measurements are used to assure also the safety and quality of food, pharmaceuticals and water. So, analytical chemist also has some role to play to the society, that you assure that the food quality is maintained. Whenever you take some drinking water, always you should know, this is basically some awareness, some basic and fundamental knowledge and information, that what amount of potassium and sodium ions you are taking with that particular drinking water, and you are not consuming by mistake any arsenate or arsenate ion from your water. Similarly, the pharmaceutical material also, you always know that whenever you have a material, whatever you are talking about, it is not 100% pure. So, some percentage of impurity is there, and what sort of impurity, even it is a dye for making the tablet or the capsule or other extraneous matter what is present over it, you should know that. And then the quality of the water, always we handle water for making food, preparing all these pharmaceutical products and food also. So, you have the corresponding restriction and the compliance with the environmental corresponding restriction what is given for the environmental protection agencies and other agencies, the regulatory authorities. So, with environmental and other regulations, so you must have these regulations that your drinking water should not contain more than 3 ppm or 4 ppm of potassium as well as sodium. So, you should follow that whenever you buy it, particularly not that water available from your river or groundwater, but when you buy it from some companies water who are making for our drinking water or the bottled water. So, they should follow these environmental restrictions, and they should print on the bottles basically whatever samples you can have. And to support the legal process, if something is happening, because the forensic science, if somebody is coming to you and needs some help, whether something is contaminated with arsenic, something is contaminated with lead or something is contaminated with cadmium, how you help it? So, the legal process can also be helped. So, the legal advisors, the lawyers, the judges all should have some very basic understanding about the analytical science, because otherwise you cannot know at what amount of cadmium you can have, what amount of lead you can have in your sample, whether it is a medicine, whether it is a food material, whether it is a water sample. So, to help the physicians also, so it is a direct application that you can help those physicians to diagnose sometimes the diseases. You know that the percentage of your glucose in the blood, then the amount of your sodium or the potassium or the calcium, these are the very simple and the very basic things what you can help other people to know, that you can determine from any such samples. So, what we get, that electrolytes, just now we are talking about that electrolytes are your sodium chloride or potassium chloride, simply we can have. So, when you go to any pathological center, the common chemical clinical chemistry they do, they go for the test to analyze the corresponding electrolytes presents. So, one such electrolyte is your chloride, because always we talk about the corresponding concentration of sodium and the potassium, but we neglect the corresponding anion of this, because all these things we take as the corresponding sodium salt. So, chlorides will automatically come if you talk in terms of the sodium and the potassium, the chloride ion, the Cl minus. So, this can also be considered as a good electrolyte trafficking in and out of the cell. So, we are talking in terms of the corresponding sodium potassium pump and all these things, but we do not talk much about the corresponding chloride concentration, because you have also the chloride channels like sodium, potassium and calcium channel, and the cell homeostasis and transmitting action potentials to the neurons due to the presence of varying chloride concentration like of your corresponding proton concentration and the proton motive force. And when we talk in terms of these different electrodes, as silver chloride electrodes are used in many applications, we know that the silver chloride electrode. So, this chloride part is coming over here. So, you get the silver chloride electrode, which is a half cell electrode we know, and such as biomonitoring sensor also is not that directly you are determining the corresponding O2 concentration or the glucose concentration or the phenol concentration, but it can be able to sense something as part of the thing from our childhood. Basically, you have been listening about the electrocardiography and electroencephalography, the ECG and EEG always we know. So, these two things are, since we are talking about electro and cardiogram is the cardiography we do. So, is basically the electron transfer, that means you must have some electrode system. So, 12 electrodes basically we use for measuring ECG. So, whenever you have some idea about the electrodes, you can also think of what sort of electrode is using for measuring your ECGs. Okay, thank you very much.